Hello everyone, I'm Dr. Arpita. A very warm welcome to my developmental biology lecture series. The topic for today's discussion is sex determination. We will do it in two parts. First, we'll talk about sex determination in mammals, and then we'll talk about the same in Drosophila. Okay, so let's get started. Sex determination in mammals is classified into two subheads: primary sex determination and secondary sex determination, right? See, primary sex determination is strictly chromosomal. Fine. Primary sex determination is strictly chromosomal, and it depends on whether the gonads form testis or ovary. That means it depends on the gonads whether it will form testis or it will form ovary. That means what? If testis is formed, it will develop into a male organism, and if ovaries are formed, it will develop into a female organism. Okay, so first see the genital ridge. Okay, this genital ridge is acted upon by SF1, WNT1 and LHX1. Okay, these factors are important to make this genital ridge into a bipotential gonad. Why do we call it bipotential? Because it has the potential to either become ovaries or become testes, right? So these steroidogenic factor, SF, WNT, LHX1, all these will form this bipotential gonad, and this bipotential gonad will give rise either to ovaries or testes. Okay. If the bipotential gonad is acted upon by DAX1 and WNT4A, it will result into ovary. That means development of female. And if it is acted upon by SRY and SOX9, it will form testes. Clear? Now, what did I say just now? It is divided into two types, primary and secondary. Primary is strictly chromosomal, and secondary sex determination is done by hormones. These hormones are secreted from the gonads. Fine. See, this ovary has two types of cells. Okay, granulosa cells and thecal cells. Fine. These granulosa cells and thecal cells, they will form follicles, and these follicles will produce estrogen hormone. Clear? Now this estrogen hormone will have this following function. These functions are very important functions of estrogen. What will estrogen do? The estrogen will act upon the Mullerian duct. The estrogen will act upon the Mullerian duct and will influence the formation of female internal genitalia. And it will also lead to the formation of female external genitalia. Internal genitalia includes what? Uterus, oviduct, cervix, upper vagina. And female external genitalia includes what? Labia, clitoris, and lower vagina. So this entire development is being controlled by estrogen here. This estrogen is doing the entire function. Clear? Now coming to testes. Testes have two types of cells, the Sertoli cell and the Leydig cell. Sertoli cell is again acted upon by this SF1, steroidogenic factor 1. Leydig cell is also acted upon by SF1. Now, this Sertoli cell will produce anti-Mullerian hormone. AMH is anti-Mullerian hormone. Fine. And this anti-Mullerian hormone will cause regression of Mullerian duct. Fine. Here you see from Leydig cell, testosterone production happens and testosterone production happens and it also produces dihydroxytestosterone. DHT is dihydroxytestosterone. Fine. Now see what happens here in males. This dihydroxytestosterone will produce or will cause this genital tubercle or urinogenital sinus to produce penis, prostrate and scrotum. Fine. And testosterone will act upon this Wolffian duct. Okay, it will act upon this Wolffian duct to develop male internal genitalia, which includes what? Epididymis, vas deferens, and seminal vesicles. Clear? So basically, testosterone is responsible for the formation of internal genitalia. That means these organs. Actually, it causes the Wolffian duct to produce all these things, and dihydroxytestosterone is responsible for formation of the secondary ones, that is penis, prostrate, scrotum, all these. Fine. Now just let's talk about few very important terms here. Each of these terms are very important. We will know the testis determining genes. What are these testis determining genes? This is SRY and SOX9. SRY is present on short arm of Y chromosome. Okay, SRY is present on short arm of Y chromosome. Okay, so SRY stands for sex determining region of Y chromosome. Sex determining region of Y chromosome. SRY. Clear? Now, another is what? Ovary determining genes. Ovary determining genes. What are they? That's 1 and WNT4. Clear? See, now you all know that XX chromosome constitution means female, XY chromosome constitution means male. Therefore, X doesn't determine what will be the sex of the child. It is the Y chromosome which plays the key role here. Okay, so I'm just telling you two conditions. Try to tell me what is the answer. If there is no short arm and only long arm, what will happen? No short arm of Y chromosome, of course. Only long arm. What will happen? No short arm means what? SRY is not expressed. What did I say? See, SRY is present on the short arm of Y chromosome. If SRY is not expressed, what will happen? It will develop into a female child since SRY is not expressed. Clear? Second condition is what? No long arm, only short arm. What will happen here? Male formation. Why? Because SRY is getting expressed here, right? SRY is in the short arm here, not in the long arm. So whether long arm is present or absent, it has got no influence on the sex of the developing organism. Right? Now, what is the mode of action? See, you should know this. This will help you to get a clearer picture of the entire concept. First is direct. What happens here, you see? SRY directly induces the epithelium. I'm writing it for you. SRY directly induces the epithelium of the genital ridge. Of the genital ridge so that it forms Sertoli cells. Right? This SRY is directly inducing the epithelium of the genital ridge so that it forms Sertoli cells. Clear? This is direct. Next is indirect. What happens here? In indirect, what will happen? SRY will induce the genital ridge to produce a chemotactic factor known as FGF9, and FGF9 will cause migration of mesonephric cells to the gonadal enlarge. Okay, actually this genital ridge has got two parts to it. See, this genital ridge has got two parts. One is the gonadal enlarge, and the other is the mesonephric cells. Fine. Genital ridge has got two, gonadal enlarge and mesonephric cells. Fine. Now, in indirect, what will happen? SRY, what did I say? SRY induces the genital ridge to produce FGF9. FGF9 is a chemotactic factor. Now, this FGF9 will cause migration. This will cause migration of mesonephric cells to gonadal enlarge. Migration of mesonephric cells to gonadal enlarge. Clear? So this is the basic mode of action of SRY. So whenever there is migration of mesonephros, testis formation will occur. And remember, if there is no SRY, there is no migration. Fine. So these are something very basic, very important and very, very basic, which you should know in order to understand the basics of mammalian sex determination. Fine. Now we'll talk a little about this ovary determining genes. What did we say? What are the two ovary determining genes? This one, and the other is, right? Okay. The nuclear receptor proteins are present on the short arm of Y chromosome. Here, sorry, X chromosome. So nuclear protein present on the short arm of X chromosome. Fine. Now, two copies of that one on the active X chromosome in XY individual. Pay attention, listen. Two copies of DAX1 on the active X chromosome in XY individuals. Now, the individual leads to formation of gonadal dysgenesis. Fine. Individual will lead to the formation, if this is the condition, that two copies of DAX1 on the active X chromosome in XY individual, then the individual leads to the formation of gonadal dysgenesis. That means what? It will result into a female individual, because see, in the XX female, one copy of DAX1 is active. In XX female, one copy of DAX1 is active. Okay. What about this WNT4? It is located on autosome. It is located on autosome. Now, when this WNT4 gets deleted, what will happen? Ovaries are small, fine, ovaries small and less mature follicles. In case this is deleted, what will happen? Small ovary and less mature follicles. Less mature follicles. Fine. So something, this is something very important. You must know what will happen if this particular gene, or I mean the protein, is absent. Fine. So this is the basics of sex determination in mammals. I hope you have enjoyed the lecture. In case you have any confusion, any doubt, please feel free to get back to me. In the next lesson, we will talk about sex determination in Drosophila. As I've said, I have divided this into two lectures. Lecture 1 is on mammals, and the second one will be on Drosophila. Now, before we end, let me tell you one important, interesting thing: androgen insensitivity syndrome. Androgen insensitivity syndrome. I don't know whether you have heard of this or not, but just for your sake of knowledge, I'm telling you. Androgen insensitivity syndrome. Here, what happens? The receptor for testosterone is absent. Receptor for testosterone is absent. Okay, external appearance of the individual is like female due to estrogen, that is sterile female. And AMH present, so no female structure is formed. AMH means anti-Mullerian hormone, right? This AMH is also known as Mullerian inhibiting hormone. Don't get confused, it is same. So for this male phenotype, you need to have AMH and testosterone. Fine. Since this AMH is present, it will not be a fertile female. So these little exception, I mean, these different conditions and all should be ready at your fingertips. Otherwise, you might lose marks in exam. So till then, thank you so much for joining the lecture. See you soon in the next one, that is sex determination in Drosophila. Till then, thank you.